Hey guys, now I'm going to continue with a brief discussion of what we would have done for fieldwork had we been able to visit Steger Creek and Rivanna River next week, and I'll be going over the types of data we'll collect and a brief introduction to how we will manipulate and analyze this data. So for fieldwork, we would have been collecting data to test the predictions of the RCC and see how well the expectations of the RCC matched up with our two streams. To do this, we would examine abiotic and biotic features of the two streams to contrast the lower order stream to the characteristics of the high order stream and see how these data compare to the predictions laid forth by the RCC. We'd look at physical attributes like stream order, stream flow, temperature, DO and discharge, and also look at aquatic productivity, which we use dissolved oxygen for as a proxy, and that can lend inferences on the water quality. And we would also look at macroinvertebrate diversity. So first, it's important to note that many streams have two major subhabitats within them, riffles and pools. Riffles are areas of shallow, fast-moving water, like this area here, and so think rapids in a river, right? And pools are areas of deep, slow moving water, like you can see here. These microhabitats can have different features like different levels of oxygen, different types of organisms living within them, different water velocity, etc. So to get a good representative assessment of the conditions of the stream as a whole, we want to capture these subhabitats and make sure that we get both of them. Therefore, for all the following data that we collect, we will make sure to include at least 2 riffles and 2 pools, which we will average together for each stream. Now, make sure to take note of the conditions between the riffles and pools too, as this leads to interesting inference as well that the RCC may or may not touch on, and you might think that we should. So overall, these are the things that we will be collecting, and field videos have been posted demonstrating these practices to our YouTube channel. So make sure you check those out. And if you listen to this lecture and it doesn't make sense, maybe go watch those videos and then revisit this part of the lecture just to be absolutely clear on what the methods are, because it's so much harder to understand this when you're not doing it yourself. And our manual as well has a really great description of these field methods. But essentially, we are measuring aquatic productivity using dissolved oxygen as a proxy, invertebrate diversity, actually it's macroinvertebrate diversity, should fix that, temperature, velocity, depth, and width. So dissolved oxygen, or DO, will be measured as a proxy for aquatic productivity. As aquatic plants and algae photosynthesize, they release oxygen into the water column. So while measuring photosynthesis in the field can be very trying and complicated, measuring DO in the water is often used as an easy proxy for productivity. And this plays a major role in determining the health of the stream. As all organisms living in the stream depend on how oxygen-rich the water is, it's a great indicator of the integrity and the water quality. To measure this, we use a DO meter. We let it calibrate, and then we dangle the probe into 2 riffles and 2 pools throughout Steger Creek and then throughout the Rivanna. We wait for the probe to settle on a consistent DO reading before we record it. In the end, we will average these for the stream, but remember, it's important to note how these change between riffles versus pools. For invertebrate diversity, or more specifically macroinvertebrate diversity, they are another good indicator of stream health, which is why we study the diversity of these little critters. As you may have learned in other classes, greater biodiversity is indicative of healthy communities and also leads to more resilient communities too. So studying changes in biodiversity can sort of be like a canary in the coal mine of disturbances disrupting the stream's health. To measure this, we use dip nets and gently disturb the substrate upstream or disturb the stream bottom, and we sieve out everything that floats downstream. We carefully pull the nets out of the water and collect the macroinvertebrates and place them in observation trays where we can use a field identification key or a guide to ID them and keep tally of the different taxa and how many individuals of each taxa we are finding. We then release them downstream from the collection site, so there's no risk of double counting. And now on Colab, there is a key posted that walks you through all the little different organisms, what certain qualifying features to look for. And on the YouTube channel, there is also a really great in-depth video on how to identify these different types of macroinvertebrates and what they might indicate for the stream. So whether they're sensitive macroinvertebrates or very tolerant, whether they're detritivores or shredders, or if they're more predatory, all of these things are very important in assessing the health of the community and therefore the health of the stream. For temperature, we take these measurements into riffles and pools as well. Temperature is important in many ecological processes, from primary production to decomposition to organism preference to ability for oxygen to dissolve into the water column or diffuse into the water column, and therefore is also important to note. So we use the DO meter, which not only measures DO but also takes temperature readings. And we dangle that probe in 2 riffles and 2 pools, wait for it to settle on a value, and then write that down. And we will average them between the 2 riffles and the 2 pools to get an average for the stream, but make sure that you're making note of the differences in temp and DO between riffles and pools. Discharge is the amount of water moving through a stream channel at a certain time or over time. It's a composition of the velocity, or the speed that the water is moving, the width of the stream channel, and the depth of the stream channel. For a representative average across the stream, or of the stream, we will actually measure 4 cross sections across the stream. So we pick 4 different locations along the Steger and 4 different locations along the Rivanna. And we make sure that they cross 2 riffle areas and 2 pool areas so that we're getting those 2 microhabitats in there. We measure the depth and the width at each of those 4 locations to get an average of the stream depth and width. And at those same 4 locations where we took depth and width measurements, we also measure velocity of the water flowing there. So we use this, or we use a bobber to do this, and we essentially drop a floating bobber like a leaf or a stick a little bit upstream and measure the amount of time it takes to float 1 meter, 2 meter, 10 meters. Your data sheets will indicate what the arbitrarily assigned distance will be. And this gives us the speed that the water is flowing in those areas. Now altogether, the speed of that water flowing, the width and the depth of the channel, these metrics will give you the discharge, which again is the amount of water moving through the stream over time. Now remember, for all of these, we're looking at riffles versus pools because they can have very different microhabitats. We will average them all, so we have an average for the stream, but it's very important to note how they differ and what this means in terms of stream health, the RCC, how valid the RCC is, etc. Watch the field videos posted, and this will make a lot more sense. And feel free to visit the PowerPoint that will be posted as well as this lecture to go over this again, but your field ecology manual also has a great explanation for these methods. So how are we going to analyze this data? For the analysis portion, we're aiming to compare and contrast the abiotic and the biotic features between our low order versus high order stream, and look at how these results compare to the predictions laid out by the RCC. Our overall goal is to test the validity of the RCC, so how accurate and how helpful we think this sort of guide to abiotic and biotic conditions based on stream order is, compared to what we found in the real world or what our data shows. You will each also make a personal or more specific hypothesis as well based on our data, which we will go over in the next slide. For our analysis, we will use this year's data, quote unquote, this year's data, as well as add it to data collected from the last few years to provide a larger data set to run statistical analyses on. We would never run a stat test or a t-test, for example, on just 2 data points, your temperature from Steger versus Rivanna. So we've collected a more long-term data set, which we will add this year's data to. Now you do want to observe how the RCC predictions relate to this year's data and then compare to the long-term trend. So keep that in mind. You're looking at what the data shows at this point in time that we collected it, and what it shows in the long-term trends as well. We will use stream depth, width, and water flow velocity to calculate the discharge for the Rivanna and the Steger. We will calculate Simpson's diversity index based on our macroinvertebrate identifications in order to provide us with how diverse the organismal community is and whether it is evenly spread or highly dominated by various taxa. This is outlined in your ecology manual and the stream intro pre-reading, so please revisit that. And I will also have a short mini lecture on that as well following this one. And then we will use t-tests to test whether differences in the temperature, DO, macroinvertebrate diversity, and discharge are actually statistically significantly different between the low order Steger Creek and the higher order Rivanna River. Remember, the RCC suggests that they should be distinct from one another. They should be different, and the RCC also suggests how they will be different. So does a low order have the higher temp, or does the high order have a higher temp? Did we actually see that in our data? And if there is a difference in the temperature, is it the way that the RCC predicted? And is it actually statistically significantly different? So we will examine whether the characteristics we found in Steger versus Rivanna follow the prediction of the RCC. So for example, if the RCC says low order streams should be colder than higher order streams, did our data reflect this? And you'll want to look at this year as well as the overall data from the long-term data set. So using the last couple of years worth of data, because those can differ. Sometimes a stream will follow what the RCC predicts, and then long-term it might not, or vice versa. And that offers important and interesting implications as to how streams might behave from year to year or seasonally. And these things are very important to note when you're assessing the validity and usefulness of the RCC or in monitoring the stream health and tracking potential disturbances over time. So the overarching hypothesis, again, is the assessment of the RCC. So will the two streams, the low order Steger Creek and the higher order Rivanna River, differ in the ways that the RCC predicts? And will these differences be statistically significant? Then you want to create your own personal hypothesis, making sure that the hypothesis can be tested by these variables that we've measured, which are listed here for your convenience. So you can use any pairing of these different kinds, or use one from a single column, or have a multiple pronged hypothesis if you like, but just make sure that the data we collect and the analyses that we will be performing can actually answer your question. For your reference, here are some indicators of stream health. And one of the major reasons that the RCC has become widely used is to offer assistance in tracking stream health. Tracing productivity, using dissolved oxygen as a proxy, and the presence of macroinvertebrates are all common indicators used to track and monitor stream health over time and assess the extent of damage following a disturbance. So in Colab, there will be a macroinvert PowerPoint as well that gets you started. And on the YouTube channel, there will be a video going over sensitive versus intolerant, sensitive or intolerant macroinvertebrates versus tolerant macroinvertebrates. So there will be pictures of several of the main ones found at Steger and Rivanna are provided at the end of the macroinvertebrate guide PowerPoint, but it's up to you to go through that PowerPoint and at the end go through and look back and determine which of those macroinverts pictured are sensitive and intolerant versus ones that are tolerant taxa. So sensitive/intolerant taxa are usually only found in pristine, healthy streams, typically involving high levels of dissolved oxygen, ample food sources, clear, clean water, tolerable pH, no pollution, etc. Whereas the tolerant taxa can exist in disturbed or distressed environments. So if you aren't finding any sensitive or intolerant taxa, and you're only finding tolerant taxa, it could be a clear indicator that the stream is suffering in some way. So again, the videos demonstrating these various field methods will be uploaded and available on our Ecology YouTube channel. Keep in mind that week 9, the week after the stream collection lab, so essentially next week, a draft of your introduction and methods for stream lab will be due. Directions for that are on page 34, and remember that you need 2 citations for your introduction. So you should be looking for papers that discuss stream quality and health, various ways to track and monitor stream quality, the importance of stream ecology and tracking stream health, anything like that, or what kind of macroinvertebrates are tolerant versus not, and try to find things that will lend support or background information for your specific hypothesis, as well as the RCC overall. Now, again, your TA might be doing things a little bit differently, so if they tell you something else, obviously ignore the slide and listen to what they say, but keep in mind that those are coming up, and the videos with all these field methods will be posted either later this week or next week, and we will have more in-depth tutorials on how to perform these data manipulation and statistical tests for you as well.